Beneath the melting ice of northeastern Greenland, an extraordinary discovery is unfolding. As permafrost thaws at unprecedented rates, scientists have unearthed microorganisms that have remained dormant for up to 30,000 years. These aren't just any microbes, they represent an entirely new branch on the tree of life, with unique abilities that could change our understanding of biology and potentially offer solutions to modern problems. Join us as we explore the Thule biome, a living window into Earth's biological past that challenges what we thought possible about life's resilience and adaptability. Our story begins in the remote reaches of northeastern Greenland, where research teams from the University of Copenhagen and the Greenland Institute of Natural Resources were conducting routine core sampling as part of a climate monitoring project. What they found was anything but routine. As the region's permafrost continues to thaw at alarming rates, these scientists identified several previously unknown microbial species that had remained frozen in time for an estimated 15,000 to 30,000 years. The microorganisms, collectively named the Thule biome after the region where they were found, represent something unprecedented in modern science, an entirely new branch on the phylogenetic tree of prokaryotic life. Dr. Elisa Magnusson, the lead microbiologist on the expedition, was the first to recognize the significance of what they'd found. Initial genetic sequencing revealed microbial signatures unlike anything in current scientific databases. These weren't simply variants of known microbes, they represented entirely novel metabolic pathways that had evolved in isolation under some of Earth's most extreme conditions. What makes the Thule biome particularly remarkable is its extraordinary resilience. These microorganisms have survived multiple climate shifts, including the last ice age and subsequent warming periods. Their genetic material provides us with a living window into Earth's biological past, offering unprecedented insights into how life adapts to extreme conditions over evolutionary timescales. The research team has identified several genes with no modern analogues, potentially representing extinct biological functions that have disappeared from contemporary microbiomes. These are essentially biological fossils, living relics of ancient biochemical processes that no longer exist elsewhere on Earth. The discovery challenges our understanding of evolutionary biology and the limits of life's adaptability. These microorganisms represent a unique evolutionary experiment, developing in isolation while the rest of the microbial world continued along different evolutionary paths. By comparing the Thule biome with modern microbes, scientists can now trace divergent evolutionary strategies for survival in extreme environments, providing new perspectives on how life might adapt to changing conditions in the future. How did these microorganisms survive millennia in a frozen state while maintaining cellular integrity? This question has captivated researchers since the discovery. The Thule microbes possess specialized membrane structures that appear to prevent ice crystal formation within their cells, a common cause of cellular death during freezing. This adaptation alone represents a significant evolutionary innovation. Even more fascinating is their metabolic versatility. Laboratory analyses indicate these microorganisms can process carbon compounds at temperatures just above freezing with remarkable efficiency. They utilize a previously undocumented biochemical pathway that allows them to extract energy from organic matter while producing minimal carbon dioxide, a process researchers have tentatively labeled cryogenic carbon sequestration. As Dr. Jens Dovelson of the Arctic Research Center explained, these microbes essentially hibernate when frozen, but upon thawing, they can immediately begin processing carbon compounds in ways we've never observed before. They're incredibly efficient at low temperatures, which could have significant implications for understanding carbon cycling in polar regions. Further analysis has revealed additional survival mechanisms that challenge our understanding of microbial dormancy. The Thule biome contains specialized DNA repair enzymes that appear to function even at near-freezing temperatures, allowing for the correction of genetic damage accumulated during their long dormancy. This contradicts previous assumptions that DNA repair processes would be effectively halted in frozen environments. The microbes also produce unique cryoprotectant molecules, compounds that prevent cellular damage during freezing, that differ structurally from those found in other cold-adapted organisms. These molecules interact with cellular membranes in ways that maintain fluidity even at temperatures where conventional membranes would become rigid and non-functional, allowing the microbes to resume metabolic activities almost immediately upon thawing. The awakening of these ancient microbes raises important questions about how they might interact with modern Arctic ecosystems as permafrost continues to thaw across the region. The research team has identified two potentially contradictory effects that could have global implications. On one hand, these microbes could accelerate the breakdown of previously frozen organic matter, potentially releasing more greenhouse gases and creating a positive feedback loop that further warms the climate. This has been a significant concern among climate scientists studying permafrost thaw. However, preliminary evidence suggests the opposite might be true. The unique metabolic pathways of the Thule biome appear to sequester carbon rather than release it as CO2. As climate scientist Dr. Sarah Rasmussen noted, we are observing something unexpected. These microorganisms seem to convert organic carbon into stable compounds that resist further decomposition. If this process occurs at scale across thawing permafrost regions, it could mitigate some of the expected carbon release. This finding is particularly significant given that Arctic permafrost contains roughly twice as much carbon as is currently in Earth's atmosphere. Understanding how ancient microbial communities interact with this carbon as it thaws has become an urgent research priority. The research team has established monitoring stations across northeastern Greenland to track microbial activity in thawing permafrost zones. Early data indicate that the Thule biome organisms spread rapidly once conditions become favorable, potentially outcompeting modern microbes in specific ecological niches. This competitive advantage appears linked to their ability to function efficiently at the temperature boundaries between frozen and thawed states, precisely the conditions becoming increasingly common in the warming Arctic. Beyond climate science, the discovery has generated tremendous excitement in biotechnology circles. The unique enzymes and metabolic pathways identified in the Thule biome function efficiently at temperatures where most biochemical processes slow dramatically. This cold-adapted biochemistry has immediate applications across various industries. Biotechnology firm Cryoenzyme has already filed provisional patents on applications using synthetic versions of these enzymes for low-temperature food processing, pharmaceutical manufacturing, and bioremediation in cold environments. Dr. Hendrik Voorman, the company's research director, believes these enzymes could revolutionize industrial processes that currently require energy-intensive heating. The ability to conduct enzymatic reactions at near-freezing temperatures could significantly reduce energy costs across multiple industries, Voorman stated. These microorganisms have evolved mechanisms to degrade complex organic compounds under conditions we previously thought too extreme for efficient biological activity. The pharmaceutical potential is particularly promising. Several compounds produced by the Thule microbes show antimicrobial properties against modern pathogens, potentially representing a new source of antibiotics at a time when antimicrobial resistance threatens global public health. One compound, provisionally named thulecycline, has demonstrated effectiveness against several multi-drug-resistant bacterial strains in laboratory tests. This could open new avenues in the fight against antibiotic-resistant infections. Environmental remediation represents another promising application. The microbe's ability to metabolize complex carbon compounds at low temperatures makes them ideal candidates for cleaning up contaminated sites in cold regions, such as Arctic oil spills or industrial waste in northern latitudes. Initial field tests suggest these organisms can degrade petroleum hydrocarbons at temperatures as low as 2 degrees Celsius, significantly below the threshold at which conventional bioremediation approaches become ineffective. The pharmaceutical potential of the Thule biome deserves special attention. In a world increasingly threatened by antibiotic resistance, these ancient microbes may offer new weapons in our medical arsenal. Thulecycline, the provisionally named compound discovered in the Thule microbes, represents just the first of what researchers believe could be many novel bioactive molecules. Initial testing shows thulecycline works through a mechanism distinct from existing antibiotics, potentially circumventing established resistance mechanisms. Laboratory tests have demonstrated its effectiveness against several multi-drug-resistant bacterial strains, including those responsible for hospital-acquired infections. What makes these antimicrobial compounds particularly interesting is how they evolved. The Thule microbes developed these compounds not to combat human pathogens, but likely as competitive mechanisms against other microorganisms in their ancient ecosystem. This represents a fundamentally different evolutionary pressure than the one that shaped modern antibiotic-producing microbes, potentially yielding compounds with novel mechanisms of action. Pharmaceutical researchers are particularly excited about the stability of these compounds at various temperatures. Unlike many existing antibiotics that require careful temperature control, the molecules produced by the Thule biome remain stable across a wide temperature range, an adaptation to their variable natural environment. This could significantly reduce costs associated with the cold chain distribution of medications, particularly in resource-limited settings. The environmental remediation potential of the Thule biome represents one of its most immediate practical applications. As climate change accelerates development in Arctic regions, environmental contamination from oil exploration, mining, and increased shipping presents growing challenges. Conventional bioremediation approaches often fail in cold environments because the microorganisms typically used function poorly at low temperatures. The Thule biome organisms offer a revolutionary solution. Their ability to metabolize complex carbon compounds at temperatures as low as 2 degrees Celsius makes them uniquely suited for cleaning up contamination in polar and subpolar regions. Initial field tests have demonstrated remarkable results. In controlled experiments using soil contaminated with diesel fuel, the Thule microbes degraded approximately 60% of the hydrocarbons within 3 weeks at temperatures of 4 degrees Celsius, a rate typically achievable only at much higher temperatures with conventional approaches. This cold-adapted bioremediation could transform how we approach environmental cleanup in Arctic regions. Beyond petroleum hydrocarbons, researchers are investigating the Thule microbes' ability to process other contaminants, including heavy metals and persistent organic pollutants. Preliminary results suggest certain strains can immobilize toxic metals through biosorption and biomineralization processes, effectively removing them from the active environment. Environmental engineers are particularly interested in developing contained bioreactors using these microbes for processing contaminated water and soil in Arctic communities, where conventional treatment methods are often impractical due to extreme conditions and limited infrastructure. Despite the excitement surrounding the Thule biome, researchers face significant challenges in studying these ancient microorganisms. The first challenge is methodological: how to study organisms that evolved under conditions that no longer exist. The research team has developed specialized laboratory environments that simulate the conditions of ancient Greenland, including carefully controlled temperature fluctuations, atmospheric compositions, and nutrient profiles based on paleoclimate data. Another significant challenge involves preventing contamination, both of the samples by modern microbes and, potentially, of the modern environment by the ancient ones. The research facilities working with the Thule biome have implemented stringent containment protocols, similar to those used in high-security biological research. While there's no evidence these microbes pose any threat to modern organisms, scientific caution dictates careful handling until their full properties are understood. The very age of these microorganisms presents another challenge. While their DNA has remained remarkably intact, some degradation has occurred over millennia. Researchers are using advanced computational methods to reconstruct complete genomes, comparing fragments across multiple samples to identify and correct for age-related damage. Perhaps the most profound challenge is philosophical. These microorganisms represent a biological heritage that has survived for tens of thousands of years. How should humanity approach this living record of Earth's past? Questions of conservation, commercialization, and scientific responsibility are being actively debated within the research community and beyond. The discovery of the Thule biome has catalyzed new international research collaborations. What began as a climate monitoring project has expanded into a multidisciplinary effort involving microbiologists, evolutionary biologists, climate scientists, biotechnologists, and environmental engineers from institutions worldwide. Future research will focus on several key areas. First, expanding the geographical scope of sampling to determine whether similar ancient microbial communities exist in other permafrost regions across the Arctic. Preliminary evidence suggests the Thule biome may not be unique to Greenland but could represent a widespread phenomenon across the circumpolar north. Second, researchers plan to conduct controlled field studies to better understand how these ancient microbes interact with modern ecosystems as they reawaken. This includes monitoring their spread, competitive dynamics with modern microbes, and effects on carbon cycling in thawing permafrost zones. Third, the biotechnology applications will be developed through partnerships between academic institutions and industry. Cryo-enzyme is just one of several companies now working to develop practical applications from the Thule biome's unique biochemical properties. Finally, the discovery has prompted a re-evaluation of planetary protection protocols for astrobiology missions. If microorganisms can survive for tens of thousands of years in Earth's permafrost, similar organisms might potentially survive in the permafrost of Mars or the icy moons of Jupiter and Saturn. This has implications for both the search for extraterrestrial life and the prevention of contamination by Earth organisms during space exploration. The awakening of microorganisms that have remained dormant since before human civilization raises profound philosophical and ethical questions. These living organisms represent a biological time capsule, a direct connection to Earth's distant past that predates written history. They challenge our understanding of life's continuity and the boundaries between past and present biospheres. Ethicists and indigenous rights advocates have raised important questions about the ownership and stewardship of these biological resources. The Inuit communities of Greenland have traditional claims to the land where these discoveries were made, prompting discussions about benefit sharing and indigenous involvement in research decisions. The Greenland Institute of Natural Resources has established a collaborative framework that ensures indigenous perspectives are incorporated into research governance. There are also broader questions about commercialization. Should organisms that represent Earth's biological heritage be subject to patents and commercial exploitation? The research consortium has adopted a balanced approach, with certain fundamental knowledge remaining in the public domain while specific applications may be developed commercially with benefit-sharing arrangements. Perhaps most profound is the question of our responsibility toward these ancient life forms. As Dr. Magnuson reflected, these microorganisms have survived multiple climate transitions over tens of thousands of years. They represent a form of biological heritage that belongs not just to science or industry, but to humanity as a whole. Our approach must balance scientific inquiry, practical applications, and respect for the remarkable biological legacy they represent. The Thule biome discovery illustrates an important paradox of our changing climate. While climate change poses serious threats to ecosystems worldwide, it is also revealing previously inaccessible biological resources and scientific insights. As Greenland's permafrost continues to thaw, a process that raises serious environmental concerns, it simultaneously unveils these ancient microbial communities that may offer solutions to some of our most pressing problems. This doesn't diminish the urgency of addressing climate change, but it does highlight the complex relationship between environmental change and scientific discovery. The discovery also emphasizes how much remains unknown about Earth's biological diversity. If entirely new branches of the tree of life can remain hidden in permafrost for millennia, what other biological treasures might exist in unexplored environments? From deep ocean trenches to subterranean ecosystems, our planet likely harbors countless undiscovered organisms with unique adaptations and potential applications. As Dr. Sarah Rasmussen noted, the Thule biome reminds us of how limited our understanding of Earth's biological diversity really is. We've barely scratched the surface of what's out there, and each new discovery has the potential to transform our understanding of life itself. The scientific community remains cautiously optimistic that this unexpected consequence of climate change might yield valuable scientific insights and practical applications while deepening our understanding of Earth's biological history. The discovery reinforces a fundamental principle of science, that nature has already solved countless problems through evolutionary processes, often in ways we have yet to imagine. The discovery of the Thule biome represents one of the most significant microbiological findings in decades. As Greenland's permafrost retreats, revealing microorganisms frozen in time for up to 30,000 years, we gain unprecedented insights into Earth's biological past and the extraordinary adaptability of life. These ancient microbes challenge our understanding of evolutionary biology, offering glimpses of biological processes that have disappeared from the modern world. Their unique adaptations, from specialized membrane structures that prevent freezing damage to efficient carbon processing at near-freezing temperatures, represent biological innovations with profound implications for biotechnology, medicine, and environmental science. As we face growing challenges from climate change to antibiotic resistance, these ancient organisms may offer unexpected solutions. Their ability to sequester carbon rather than release it as greenhouse gases could mitigate some effects of permafrost thaw, while their novel antimicrobial compounds open new possibilities in our fight against resistant pathogens. The Thule biome reminds us that Earth's biological diversity extends not just across space but through time. In studying these living relics of Earth's past, we gain not only scientific knowledge but a deeper appreciation for life's resilience and the biological heritage we share with all living things. As Dr. Magnusson observed, nature has solved problems we haven't even properly formulated yet. In the thawing permafrost of Greenland, we may have found answers to questions we're only beginning to ask. Thanks for watching. Please like, subscribe, and hit the bell icon for updates. Your feedback is appreciated. We hope to see you again.